Glucose metabolism induces mono-ubiquitination of histone H2B in mammalian cells. By
Rosemary Darko,
Kearney,
and Monica Sen.
The aim of this presentation is to review the original paper that reported the glucose concentration in mammalian cells induces mono-ubiquitination of histone H2B. Protein modifications are essential for transcriptional regulation and biological processes. Histone modifications occur at the tails of histones. These modifications affect chromosome function, which in turn may alter the electrostatic charge of the histone, leading to a structural change, thus a change in the ability of the histone to bind to DNA. This research paper focuses on mono-ubiquitination of histone H2B in mammalian cells. The two types of glioma cells used in this experiment were U87 and U118. The cell cultures, treated or non-treated, were harvested by scraping in the medium on ice and then placed in a centrifuge to separate the cell components. The cell pellets were resuspended in 2 SDS protein sample buffer containing beta-mercaptoethanol and heated at 100 degrees Celsius for 5 minutes. The total protein extract was separated by 12% SDS polyacrylamide gel electrophoresis and used for Western blot analytes.
Glucose nutrient deprivation inhibited mono-ubiquitinated histone H2B in cultured glioma cells. The U87 and U118 cells were first cultured in DMEM supplemented with 10% FBS. The samples were then placed in phosphate-buffered saline to remove any FBS or DMEM. The cells and phosphate-buffer saline were scraped off the petri dish and centrifuged at 300 relative centrifugal force for 2 minutes. The sample was boiled in SDS page sample for Western blotting. An antibody that was raised specifically against ubiquitination histone H2B at lysine 120 was used in the gel electrophoresis to measure the levels of ubiquitination. The samples were then incubated at 37 degrees Celsius for 1 to 6 hours with samples of the cells being collected and the results recorded. In order to discover if the levels of ubiquitination could be recovered after incubation, cells were then added to glucose-rich DMEM and the UH2B was measured over a period of 18 hours. In Figure A, it can be seen that the levels of ubiquitination of the histone H2B decreases in the U87 cell sample when added to a glucose-deprived medium. The levels of ubiquitination of the H2B histone in the sample of U118 cells also decreases significantly when added to a glucose-deprived medium. In Figure B, the levels of ubiquitination of the H2B histones in both the U118 cells and U87 cells had begun to recover after 2 hours in the glucose-rich DMEM and FBS medium. This shows that glucose-containing DMEM supplemented with FBS contained signal molecules that is required for inducing the UH2B in cultured glioma cells.
This method was used to see if glucose is an inducer of UH2B in glioma cells. We sought to identify which component in the high glucose DMEM FBS is responsible for inducing mono-ubiquitination of histone H2B in cultured glioma cells. The cells were treated for 6 hours with PBS and were placed in glucose-minus DMEM medium with either regular or dialyzed FBS. FBS contains a very low level of antibodies and contains more growth factors allowing for versatility in many different cell culture applications. This allows the cells to stay alive during the experiment. Sample A contains cells that were placed in glucose-lacking DM, EM and PBS for 6 hours with 10% undialyzed FBS. Sample B contains cells that were placed in glucose-lacking DM, EM medium and PBS for 6 hours with 10% dialyzed FBS. And sample C were placed in glucose-lacking DM, EM and PBS for 6 hours with 10% dialyzed FBS and 0.045% glucose. In sample C, U87 and U118 cells were grown in regular high glucose DM-EM media and then rinsed with PBS to remove glucose for 6 hours. Cells were then collected and replated with glucose minus DM-EM with dialyzed FBS and 0.5 grams per litre glucose. These results indicate that glucose is actually an inducer of UH2B in glioma cells. From the Western blot in sample A, this suggests that histones were mono-ubiquitinated. However, in sample B, mono-ubiquitination did not occur. This is because the dialyzed FBS may have moved small molecules such as glucose metabolites which were present in the FBS. Sample C was used to test whether glucose is definitely responsible for ubiquitination in glioma cells. UH2B was recovered after glucose incubation, hence glucose is definitely required.
This experiment showed that glucose is the component in the DM-EM FBS that is responsible for inducing mono-ubiquitination of H2B at lysine 120 in human glioma cells. However, the glucose-deprived media was defective in inducing UH2B. This proved that the glycolytic regulation of UH2B was conserved in human glioma cells.
We hope you've enjoyed our presentation. Thank you for listening.